Dear chairs, dear audience, in this presentation I would like to present my work, what I did in the last year in the BRC. First, I would like to introduce our experiments regarding the permeability of the blood-brain barrier and also the tests that we made with nanoparticles which could be used as future carriers for drugs. But first I would like to say a few words about what the blood-brain barrier actually is. The blood-brain barrier consists of 3 cell layers: brain endothelial cells, pericytes, and the end foot of astrocytes. The blood-brain barrier is a dynamic interface which is responsible for the communication between the central nervous system and the periphery. And also it maintains the homeostasis with the brain with providing the sufficient nutrients to it. But the blood-brain barrier also acts as a defense system against toxic insults which can cause pharmaceutical problems for us. But what are these problems? For example, there are neurological diseases, such as Alzheimer's or Parkinson's, which can be cured efficiently because the blood-brain barrier prevents the drug entry to the brain. And now the question is, how can the blood-brain barrier block drug entry to the CNS? First, I would like to introduce the open pathways at the blood-brain barrier, which are important for us because they can be used for drug delivery. The first open pathway is the pathway of lipid-mediated free diffusion, which allows lipid-soluble molecules to pass through the blood-brain barriers, for example, nicotine, ethanol, or other drugs of abuse. The second open pathway is the pathway of solute carriers, which are responsible for the transport of nutrients, for example, the GLUT1 transporter, which transports the main nutrient of the brain, glucose. And the third open pathway is the pathway of receptor-mediated transcytosis, which is responsible for the transport of peptides or proteins such as insulin or transferrin. And now in this slide I would like to introduce you to the closed pathways at the blood-brain barriers which are causing problems for the drug delivery to the central nervous system. The first closed pathway is closed because the presence of the tight junctions, so the paracellular traffic is inhibited. With this inhibition, the water-soluble molecules and the water itself can't cross the blood-brain barrier by paracellular method. The second closed pathway is the pathway of efflux transporters. These transporters can block the entry of their ligands, such as xenobiotics. The third closed pathway is the pathway of non-specific vesicular transcytosis, which ensures us that there is no plasma protein leakage at the blood-brain barrier, which is an important fact for us because we used albumin in our experiment, which I will say a few words about later. So, our main goal was to find several strategies to circumvent the barrier function of the blood-brain barrier. There are a lot of strategies, but we can divide them into 3 main groups. The first main group is circumventing the BBB itself by using direct delivery or the nasal pathway, which is unfortunate. Direct delivery is unfortunate because we only can enter our drugs to the ventricles of the brain, which is very invasive, and the risk of perioperational infections are very high. With the nasal pathway, we can only reach the pre-brain because we use the olfactory axons for transport. And we also have the option of modification of the BBB functions, for example, by using small peptides which can open the tight junctions and with that allow the small water-soluble molecules to go through the paracellular pathways. Or we can use P-glycoprotein inhibitors, which are efflux transporter inhibitors. Or there is a third way targeting the physiological transporters on the blood-brain barrier. This is what we used in our experiments with targeting or nanoparticles with ligands. During our experiments, we used the triple co-culture models, which look like the following. As you can see, our models consisted of an upper and a lower compartment, and we had a membrane in it. On the upper side of the membrane there were brain endothelial cells, on the lower side there were pericytes, and on the lower compartment there were the astrocytes. So with this we could mimic the functional anatomy of the blood-brain barrier. In our experiments, our first sample were an iosome, which was 100 nanometers in diameter, contained Evans blue albumin. Like I said, we used albumin as a cargo, and glucose analog for targeting ligand. Our first and most important question was whether our nanoparticles toxic or not to our cells. As you can see, we carried out toxicity tests in which we used the control and the negative control too. As you can see in the decrease of the graph of the negative control, the tritonics, which was our negative control, caused cell death. And as you can see in the long-term test, only the 10 milligram per milliliter concentration caused harm in the cells. This was our first test, what we did with non-targeted ligands, but we also tested or targeted nanoparticles too. In this test, we came to the same conclusion that in long term, only the 10 milligram per milliliter concentration caused harm to our cells, the other concentrations were fine. Then we carried out an uptake test. The method was the following. We introduced our particles into our compartment, which could enter the endothelial cells. As you can see, with this experiment, we did a parallel experiment with a targeted and a non-targeted nanoparticle. As the graph shows, the non-targeted nanoparticle could enter the brain endothelial cells, but the efficiency of the targeted nanoparticles were much more higher. To verify our results, we used confocal microscopy because the Evans blue albumin is red under confocal microscopy. So as you can see in this picture, the albumin and our nanoparticles could enter our cells. Then we carried out permeability tests. The method was the following. We introduced our nanoparticles into the upper compartment, which had to pass through first the brain endothelial cells, then our membrane, then the pericytes, and then we measured them back from the lower compartment. As you can see on the graph, in this case too, the non-targeted nanoparticle could pass through our model blood-brain barrier, but the permeability of the barrier for the targeted glucopyranose targeted nanoparticle was almost 3 times as high. Then we introduced our second sample, which were solid polystyrene nanoparticles with the diameter of 40 nanometers. Our nanoparticles were also fluorescent this time too, and they were labeled with neutrovidin. And in this case, we carried out 3 parallel experiments. In the first one, we used non-targeted nanoparticles. In the second one, we used nanoparticles targeted with biotin. And in the third one, we used nanoparticles with biotinylated glutathione targeting. As you can see on the graph in this uptake test, the uptake for the biotinylated nanoparticle was 2 times as high as the non-targeted one, but the most efficient one in this case, similarly to the previous ones, was the nanoparticle with the biotinylated glutathione targetage. We also verified our results in this case too. As you can see, as I said before, our nanoparticles were red under confocal microscopy, and they are here marked with red in our cells. We also carried out permeability tests with the same method as I showed before, in this case too, with the same 3 nanoparticles, and as you can see on this graph, in this case too, the biotinylated glutathione targeted nanoparticle was the most efficient one. So as a summary, I would like to emphasize the fact that the blood-brain barrier is crucial for nutrient transport and brain homeostasis, but it also can block the pathway of drug entry to the central nervous system. But we came to the conclusion that targeted nanoparticles are able to enter the blood-brain barrier and cross it, and they could be used in the future as possible transporters for drugs. And I would like to say a special thank to my mentor and my co-mentor, Dr. Mária Deli and Dr. Silvia Veselka, and also the University of Szeged and the Foundation for Future of Biomedical Sciences in Szeged. Thank you for your attention.